Good morning, good afternoon, good evening, wherever you are. It's a great pleasure for me to welcome you to this latest webinar provided by the International Society of Antimicrobial Chemotherapy in conjunction with the Asian Pacific Society for Clinical Microbiology and Infection. We've got a wonderful panel of 3 speakers today talking about some very interesting infectious diseases that are particularly of interest in Asia, but of interest to everybody else around the world because of the small world we live in these days. My name is David Jenkins. I'm the current president of the British Society for Antimicrobial Chemotherapy and also a council member of the International Society of Antimicrobial Chemotherapy. ISAC was founded in 1961. Sorry, my camera's gone. ISAC was founded in 1961 and is a federation of affiliated member societies whose aim is to increase the knowledge of antimicrobial chemotherapy and combat antibiotic resistance around the world. It's got a worldwide membership of over 100 national and regional societies, and they in turn have a sum total of over 60,000 individual members. One of the important activities of ISAC is to host the International Congress of Antimicrobial Chemotherapy. This happens every other year, and this year it's going to be held in early November in Istanbul, and we will be putting up links to the meeting at the end of the webinar. But I would very much encourage everyone watching today to look at the program, and we would be delighted to welcome you in Istanbul later on this year. Apart from, I will be moderating the sessions, and I will be joined by my co-host, Professor David Lai, who I will introduce. Well, who will introduce himself now. David, over to you.
Thanks, David. I'm David Lai, president of the APSCMI Society of Clinical Microbiology and Infection. I'm from Singapore. I'm an infectious disease doctor. It's a pleasure today that we are co-hosting this webinar with ISAC. Essentially, we are building on the theme of One Health, which we believe is a source of SARS-CoV-2, but we don't want to talk about SARS-CoV-2 because it's 2024. So we thought we'll talk about AMR, we'll talk about SFTS, and of course, Nipah virus. So it's my pleasure to introduce the first speaker, Professor YongHong Xiao, who is an old friend. Pre-pandemic. He's the Vice Director of the State Key Laboratory of Diagnosis and Treatment of Infectious Diseases at the First Epic Hospital School of Medicine, Zhejiang University, China. His major interest has been in various aspects of infectious diseases, but particularly in antimicrobial agents and resistant, pharmacology, and stewardship. He's the founder and leader of China Ministry of Health National Antibacterial Resistant Investigative Net. He's conducted he's conducted the first One Health AMR program in China in 2015, and has published more than 500 different papers and 30 book chapters. He will share with us today about One Health AMR program in China. Over to you, YongHong.
Okay, dear Professor Lai and Professor Jenkins, and dear colleague, good evening and good morning, I think everybody from different regions of the world. Today, I'd like to talk something about the AMRs in China, especially some concept of One Health be used for the control of AMRs. My presentation will contain the following sessions. First of all, I'd like to talk something about the AMR epidemiology in our country. As you know, AMR is a real series of problems around the world, also in China. From this figure, you can see during the past several years, the major resistance in our country have some trend of decrease. But you can see in 2011, the resistance rate for many bacteria increased to a top. For example, in 2011, the ESBL positive E. coli is around 70% of the isolates. And here you can see also MRC is over half of the isolates is MRC. And after that, because at that point, our country take a national special campaign to improve the antibiotic ration use in healthcare setting and also improve the control of AMRs, especially promote the nosocomial infection control in hospital. So after that, we get a great achievement in antibiotic use and also AMR control. You can see from that point on, most of the resistant decreased sharply. Especially here, you can see the ESBL positive E. coli decreased from 70% to around 50%, 20% decrease. And also here, you can see the MRC decreased from over 50% to around 30% here. And now the major challenge, a major problem is a slowly increase of the carbapenem-resistant Klebsiella pneumonia in our country. The nationwide, the average epidemiology is around 10%, but in some larger hospital, maybe epidemiology of the carbapenem-resistant near around 30%. Some hospital, maybe some special unit, for example, the ICU, over half of the isolated is carbapenem-resistant. Here's demonstrated some as you know, 3 major resistant bacteria in the paper. One is carbapenem-resistant Enterobacteriaceae, and here the second one is a carbapenem-resistant Pseudomonas aeruginosa, and the third one is a carbapenem-resistant Acinetobacter baumannus. And the highest resistant rate occur in Acinetobacter baumannus, around 50% around the country, and followed by Pseudomonas aeruginosa, which is around 20% of the isolates. Here I already told you, carbapenem-resistant Klebsiella pneumonia is around 10% around the country. And also, as you know, China is a wide territory country, and the resistant situation is some difference between different area. For example, most of resistant occur in the east part of China. This area always is economic development areas, and especially, for example, in Beijing, Shanghai, and also my working place, Hangzhou, the Zhejiang province, the resistant situation is relatively serious. If we focus on the carbapenem-resistant Klebsiella pneumonia, we can divide it 2 periods. Before 2011, the resistant rate of carbapenem-resistant Klebsiella pneumonia is less than 10%. But after that, the quick increase of this agents, this bacteria. In 2017, the tertiary hospital, because this is focused on the tertiary hospital. The tertiary hospital on over 20% of isolated be carbapenem-resistant. Last year, we found carbapenem-resistant Klebsiella pneumonia have a slight decrease around, for example, some hospital already less than 20%. It's a good news, I think. And except the healthcare resistant, also in our country, we found some problems that resistant bacteria could be spread between different sectors, for example, from animal to human beings, and also you can find they spread from environment to the human being or animals. So we did some study of that, get some information about the intersectoral spread of AMR bacteria. 3 years ago, we conducted a pilot study of AMRs spread in a county. This county is a sensitive population, and also the animal farmer is also very common. So we set the county as our investigational site, and we take some villages as do the study. We collect a sample from different sectors. For example, we collect sample from the health person, the farmers, animals, environment, include the water, sediment, and some others. And also sometimes include the bacterial isolate from the hospital. After that, we check in the sensitive data and also do the resistant mechanisms and also some molecular epidemiology. From this study, we can find the ESBL positive is very common in different sectors, include animal, environment, and also human beings. Here you can find the ESBL positive E. coli is around average, the level is 38%. And housed in pig, the 73%, followed by river water and river sediment and human waste, wastewater, and so on. Except the resistant to cephalosporins, those isolates also have some resistant rate to colistin, except the human isolate, the colistin resistance is 0. But the isolate from other sectors, for example, environment from animals, they connect, they E. coli keep relatively high resistance to colistin. For example, here is a bird facial isolate is resistant over 20%. And also can find some carbapenem resistant from animal from the river west. It's a big challenge, I think. Also, we check in the ESBL typing. Most common ESBL from human beings is CTXM15 and the 6XM55. And also the CTXM55 is also very common, not only in the humans, but also from medical, also from the pig, and also the water and some others. So that means the resistant genes maybe have a intersectoral spread. After that, we do the whole genome sequence of the isolate. I found it's true that the same ST type, for example, ST10, could be found in different sectors, include the human beings, bigger, and also the wastewater is a big. I think that means the ST type ST10 could have some spread between humans, animals, and the environment. Except the ESBLs, we also check the NDM. You can see in this county, the highest prevalence of NDM we found in the pig is over 10%, followed the cattle and the fly, and also the chicken and the dog. The human beings prevalence is around 2.3%, so highest in pig. And those isolates also resistant to colistin and also cephalosporin. Even more resistant rate, the human beings is higher than animals. For example, the imipenem, meropenem resistance around 100% from those isolates, but only 70% resistant rate be detected in animal isolate. And also, we check if there are some intersectoral spread. We do the MSD typing and the whole genome sequence. We found, for example, the human ST typing could be found in pig, in chicken, and also in dog. And here, for example, the major pig ST typing of the bacterial isolate could be found in the human beings, also the chicken. And also the chicken ST typing could be found in pig and also the human beings. That means that the spread in different sectors is confirmed. After that, we also check the MCR1 because at times MCR1 be first detected in China. We found in our mink farmers that we isolated colistin-resistant E. coli from both mink and the farmer. And furtherly, we detected this is MCR1 positive E. coli. And also the ST type is difference between the human and the mink isolates. But both ST type contain MCR1 and also the MIC almost the same age. After that, we check in the plasmid. We got 2 new plasmid from the MCR1 E. coli. And except the MCR1, you can find some other resistant genes be contained in the plasmid, for example, the OPX, OPXB, OPXA, and also the CTXM14. And here we found a new plasmid. This plasmid is almost the same as some other originated plasmid being resistant to colistin. except the E. coli, we also have some study of the MRC. As you know, MRC also have a sharp decrease in the healthcare setting, but there are some changes in MS typing. From 2014 to 2019, the 6 years, you can see the major typing in 2040 is ST5 and ST59. But after that, ST5 decreased greatly and ST49 increased greatly. Very surprised that ST398, the major coming from the animals, you can see could be founded and increase the isolate from the bloodstream infections from human beings. So it's a very, very dangerous situation. And also most of the animal originated MRC come from different regions in our country. And also, as you know, everybody know ST9 MRC is major infected animals, but it also got this isolated from bloodstream infection of the patient. Also, we cannot confirm the origination of these strains, but I think maybe there's some spread between animals and human beings of AMRC. The third one is the Clostridial differences. The CD is not so common in China. Most of the CD infections, CDI, is hospitalized patient after the use of the broad spectrum antibiotic. But recently, there's some real case of the communicable occurring infections. So we hope to get some information about the spread of these bacteria between animals, environment, and human beings. So we selected a city. This city is around 100 kilometers away from the Hangzhou. We collected the isolate from animals, farmers, environments, and also we collect isolate from human beings in our hospital and do some comparison of the whole genome sequence and also the MS typing. After whole genome sequence and do the MS typing, we found there's some special clone groups, CG. These CG groups could be shared by different sectors. For example, the farmers can share the same CG groups with pig, with environment, and also the patient can share the same ST group for the carriers and also for the environment of the ICU. That means maybe we speculated that the bacteria could be get in the health person, and if the person get a disease, and also could be transferred to a hospital, and then they could be spread in the hospital. So after with that, we get some information from E. coli, MRC, and also the CD. That means generally sometimes the bacteria could be circle in animal world. But if the human beings interrupt into the animal circles, we can gauge the bacteria. And also the resistant bacteria could be transferred by some other way, for example, drinking waters, wastewaters, and also fly could be as a medium to the transmission of the resistant bacteria. So after we got so many information, and also as you know, recently our countries make 2 versions of national action plan to contain AMR. In 2016, just before the G20 summit in China in 2016, 1 week before the G20 summit, the Minister of Health collaborated with other 12 ministries in our country to launch the first version of national action plan. From this purpose of the national plan, you can find the One Health concept will already be integrated into the plan. For example, here, we should realize the prescription sales of antibiotics in whole country in healthcare. And over half of the animal sales of antibiotics should be with veterinaries prescription. And also we should establish the optimizing the surveillance networks in both healthcare and animals. And also we should establish the antibiotic evaluation system in healthcare system, and also the animal has batteries. And also we should control the antibiotic resistance in healthcare setting, and also animal AMRs should be contained. That means in the first version of the National Action Plan, the One Health concept already integrated the whole plans. It's very, very important. After 5 years in implementation of the first version action plan, in fact, we achieved some success. Of course, some target we cannot meet with their target. So in 2022, the second version of national plan will be launched by Minister of Health and some in collaboration with other ministries in our country. Also, in the second version national action plan, you can see the personal, veterinaries, environmental AMRs already be included as a whole entity. So you can see the resistant control and the antibiotic ration use and some other new methodologies to control of the resistant already be described in the second version of the AMR national action plan. For example, the personnel and the veterinarians, animal use and old sales, and so on. That means One Health concept will already be used in our country. Finally, I'd like to something about our One Health practice in the control of the AMRs in our country. If we look back our history of AMR control in our country, we can divide it into different periods. And here should be the key point, 2011, because here the National Health Commissioner launched the National Special Campaign to contain the AMRs and to improve in the antibiotic ration in the hospital. Especially during the 3 years from 2011 to 2013, the administration of the use of antibiotic in the hospital is very, very strict. That means the Minister of Health setting the target for the hospital and sending the inspected team to the hospital to check the implementation of the antibiotic use of antibiotic control in the hospital. After trying a promotion of the antibiotic ration use in our country, the hospital, we can find the antibiotic consumption in the hospital decreased and also resistance situation get better. After that, the Minister of Health asked hospital establishing the antibiotic stewardship program in the hospital, and they continue to promote the ration use of antibiotics. So you can see right now, almost every hospital, the tertiary and secondary hospital, has only AMS working team to promote the ration use of antibiotic and also control AMR in our country. Here, I'd like to something our panel study in the county. After we get the information of the resistant bacteria spread in different sectors, we take some measures to control resistance. For example, in the hospital, in the villages, in the farmer, in the general.
Hi, David. I think the presentation has frozen there. Certainly has for me. Has it for you as well? Professor Xiao, you back online?
I think he has wait for him to come back. He's just Hi, Professor Xiao. We lost you temporarily. Are you back? Yeah.
We can't hear him though.
Okay, can you hear me?
Yeah.
Yes, better.
Okay,
Good.
we can hear you now. Thanks.
Thank you. This is some technical problem. Here, after 6 months, you can see the consumption of antibiotics, the animal feed decreased, and also the resistance of bacteria isolated from animal also has slightly decreased. Especially can found the general practitioners use antibiotic decreased sharply. For example, especially antibiotic use intravenous decreased very obviously. So we got a primary success in that county. And another important report is that, as you know, in 2016, we found a plasmid-mediated resistance of colistin. After that, the Minister of Agriculture banned the use of colistin in animal as the growth promoter. So after that, you can see the colistin residue in environment of animal environment decreased, for example, the chicken, the pig, and also the MCR1 decrease in the chicken and the farmer, and some provinces is also have obviously decrease of MCR1 resistant. It's good news. And 2 years ago, some research from Shanghai Chosun University conducted a study. They generated a One Health Index for the AMRs, use the global One Health Index. In the index, include 5 factor indicators. The first is AMR awareness. The second one is AMR laboratory networkers coordination capacity. The third one is AMR control and optimization. The fourth one is improving awareness and understanding. And the fifth is antibiotic resistance rate for important antibiotics. In the systems, over 30 indicators we integrated. And after creating the index, they evaluate the AMR's situation around the world. 146 countries were calculated. You can find in the full group of the World Bank economy situation, the high income countries always have a very high score in AMR control, followed by upper middle income country and low middle income country. And always the low income countries have a poor score for the AMR control. In Asia, you can see the first one, the top is Japan, followed by South Korea and China, and also here Australia. Almost the Japan score is around 60 point, and China is around 55 point, similar with the United States. Here is the same colors. And in the 5 BRICS countries, the China and Russia almost the same, and China is the number 1 in the overrank. It is 23rd of the 146 country, and Russia is 31 in 146 countries. And last week, I read a new report about the comprehensive assessment environment mission of veterinary antibiotic also in our countries. You can see the curve from 2010 to 2020, the almost 10 years, we can divide into two periods. From 2010, antibiotic consumption and emission of animal antibiotic increased to the top around 2015. After that, the emission of antibiotic from the animals decreased. And also the major emission is the tetracycline, sulfalamide, and also transuranic and some quinolones. And also here, from 2010 to 2015, there is an increased trend, but after that, the emission of antibiotic from animal feed decreased sharply. So I think from the animals, healthcare, and also some other information tells that the antibiotic consumption in our country is getting down, and also resistant situations also getting better. But I think we still have a very high resistance rate in the hospital settings. And in fact, in our countries, Minister of Health, Minister of Agriculture, and Minister of Environmental Protection already integrated cooperatively to have some actions, already made some acts or regulations in AMR control. For example, except the 2 versions of National Action Plan, there are some other very special regulations for the control of the resistant, and also banning the emission of antibiotic to the environment. I think maybe in the future we can get some more successful achievement. Anyway, AMR is big problems around the world. China is a big consumption of antibiotic, and also we have a relatively serious problems, AMRs. During the past 10 years, China already taken some measures to control AMRs, and not only in the healthcare, but also, for example, animals and environment. We already do something with One Health concept strategies in our country. With the One Health strategies, we already get some success in the antibiotic residues and also emission of antibiotic environment, and also the resistant get better in our country. But anyway, I think in the future, so we should do more in cooperation with different minister and different departments in the control of AMRs, use One Health concept. Okay, my presentation will be here. Thank you for your attention.
Remarkable work in China from the One Health approach. There's one question from an anonymous attendee. He or she asked that the lower rates of carbon resistance in NDM isolates from animals, does that mean that they reflect different NDM values compared with those from humans? Based on your whole genome sequencing, did they, are the human NDM closely related, or are they different from the animal NDMs? I think that's the first question. You have any quick answer to that?
It's very interesting. Yeah, quickly I will have a response for the question. Yeah, we did some study of the NDM1, the resistant from both animal and human beings. We compared the whole genome sequence and also compared plasmid. It's true that we found some similarity between human resistant isolate and also the animal resistant strains. I think there's some spread between the human beings and animals. Anyway, I think maybe some more research should be done, because as you know, the major resistant to the carbapenem resistant in clinic is KBC, not anymore in your country. Okay.
Thank you. And then in the interest of time, perhaps Prof. Xiao, if you could answer some of the other questions in the chat, if it's possible, thank you, and to stay behind for if you have more time for questions later. I'll pass over to David Jenkins now to introduce the next speaker. Thank you, David.
Thank you, David. The next speaker is Professor Dongmin Kim. Professor Kim is in the Division of Infectious Diseases at Chosun University in the south of Korea and Chosun University College of Medicine. His research interests include vector-borne infectious diseases such as scrub typhus, rickettsiosis, severe fever, and thrombocytopenia syndromes, and Lyme disease, as well as neglected infectious diseases including Japanese encephalitis virus, dengue virus, malaria, and parasitic infections. Currently conducting research on the development of diagnostic methods for infectious diseases, therapeutic agents, and vaccines. And Professor Kim will be presenting on severe fever with thrombocytopenia syndrome in Korea. Thank you very much, Professor Kim.
Yeah, thank you. I'm honored to present SFTS in Korea at APSCMI webinar. In the first half, I'd like to present case report, and second half, I will present clinical research I am currently conducting. 84-year-old female, complaint is fever and diarrhea. Tick was found to attach. Lab results show that leukocytopenia, thrombocytopenia, and elevated AST level. This is a microscope that attached to her. When you perform 6-next PCR test, the tick was identified as Hemaphysalis longicornis. When you perform a real-time PCR, you can detect the SFTS virus when you use tick and serum sample. When you construct phylogenetic tree, we can identify identical SFTS virus from patient's serum and CSF, as well as ticks. SFTS is acute febrile illness caused by Bandavirus. which is called SFTS virus. Mortality rate is around 20%. It has three segments. L segments encode RNA-dependent RNA polymerase. M encodes glycoprotein GN and GC. S encode nucleoprotein and non-structural protein. It is our clinical characteristic of our cohort SFTS patients between 2013 to 2018. We enrolled 221 patients. As you can see, fatal case is more older. And fatal case has less chill and fewer myalgias. As you expected, a fatal case has more mental status change. This case is a 47-year-old male. He presents with headache, fever, chill, loose stool, 5 days prior to admission. Leukocytopenia, thrombocytopenia was detected. There is a report, sensitivity comparison between real-time PCR and conventional PCR. They said sensitivity is almost the same. But when you conduct nested PCR is the PCR. When you perform PCR, we can get first amplicon. With this first amplicon, we can do second PCR using inner primer. So performing PCR test twice result in higher sensitivity. These patients on admission, SFTS conventional PCR result was negative. But when you perform nested PCR, the result came back positive. So in first two cases, when you perform conventional PCR, the test result was negative, but nested PCR, the test result was positive. So if we perform only conventional PCR in two cases, there is chance you can miss these patients. Later two cases, on admission sample, it was positive, but follow-up sample, it was negative. So if the patients visit hospital late, in that case, there is chance it could be negative. When you perform 38 patients, SFTS patients with 122 specimens, PCR result sensitivities, when you perform conventional PCR, on 7 days, sensitivity is around less than 70%. On 14 days, sensitivity is around 50%. But when you perform nested PCR, sensitivities remained until 40 days. Company K developed SFTS real-time PCR kit. But when he verified as cutoff value 37, there are a lot of false positive results in various infectious diseases. So we feel that more accurate SFTS real-time PCR test is needed. So we developed real-time PCR with our design one. Our one is more sensitive than previous one, which is a surpass nested PCR sensitivity. There is one discrepancy between our PCR and previous KFD approved real-time PCR. In that patients, the patients was diagnosed with HFRS, hemorrhagic fever with renal syndrome. PCR was positive and rising titer was documented. When we perform using company K real-time PCR, CT value was 37. But when you use our one, there is no false positive result. When you perform real-time PCR between survival and fatal case, fatal case has a higher viral copy number. When you connect in follow-up sample in figure, a fatal case tend to increase in viral copy in follow-up samples. If the copy number is less than 10,000 copy, 95% will survive. But if the copy number greater than 10,000, in that case, almost half of the patients died. In Korea, plasma exchange were used in experimental treatment. They report if they checked plasma exchange group and non-plasma exchange group, there is no significant difference. But when they subgroup analysis, they define early plasma exchange group within 7 days. In that case, they insist early plasma has a survival benefit. But in our cohort study, when you perform checked the effect of plasma exchange group has a shorter survival group. As you know, plasma exchange group has a higher FACIT score, more complication, more ICU admission. That is because I think most of the experimental treatment can be done for severe group. So by adjusting this severity, we use propensity score matching score. So after matching, there is no difference between two groups. When you compare plasma exchange group and non-P group, after match, there was no significant difference. When you group only plasma exchange group within 7 days, in that case, there is no difference as well. So early plasma exchange group does not seem to be effective. But when you compare the steroid group and non-steroid group seems to be shorter survival time after matching score as well. When you divide it in 3 groups, non-steroid, early steroid, less steroid group, early steroid within 5 days and shortest survival time. When you divide it according to severity, a FACIT score less than 14 means mild or moderate severity. In that case, if they receive a steroid, in that case, survival rate is much lower, shorter survival time. So when the patients with mild moderate severity, in that case, steroid should be very cautious. From now on, I'd like to introduce our vaccine and therapy monoclonal antibody test. Before COVID-19 pandemic, I was interested in only diagnostic test developed. But after the COVID-19 pandemic, I compared to make a contribution. So I started to do a vaccine and monoclonal antibody study. At first, I used E. coli-based vaccine study. So based on bioinformatics, protein database, epitope analysis was initiated. And this is a DPO, post-onset symptom, 7, 11, 17. As you can see, with time goes by, rising titer can be detected. So E. coli-based MP seems to be very effective antigen. But when you make non-structural protein and glycoprotein, it did not seem to be effective. even though we make a lot of truncate and chimeric protein, antigenicity does not seem to be good. When you inject this kind of protein into mouse, when you check IFA, only NP group shows reactivity against SFTS virus. When you perform neutralizing antibody test using plaque reduction neutralizing test, FRNT50, or E. coli vaccine group did not elicit neutralizing antibody at all. So we move to plant-based SFTS vaccine platform. There is some difference between human and plant in glycosylation pattern. In plant, there is a xylus and fucus were there. So we made wild type with xylus and fucus, and we made another one with deletion of xylus and fucus. We call it delta FX. This plant seems to be very effective. So we inject this protein into mouse, whether that elicit neutralizing antibody. As you can see, plant GN elicit high neutralizing antibody, especially delta FX seems to be higher neutralizing antibody. So we use this delta FX protein. When inject into mouse, the serum is cross-reactive with various clinical isogenotypes as well. The antibody level is much higher than mammalian cell injection group. So we use virus challenge test using interferon knockdown mouse. Inoculum size is 10^3. But as you can see, this is a vehicle group, this is a GN group, this is MP, GNMP. The same to body weight, there is no effectiveness, you can see. But in GN group, there is a little bit low viral copy. So I'd like to know why this plant vaccine does not seem to be effective. So we checked serum with PRNT level using serum sample taken immediately prior to virus challenge. As you can see, PRNT level is very low compared to previous our experiment. So we checked SFTS phase. This is a VSA 5 microgram. This one is the GN SFTS gram. The band thickness is significantly different. So which show there is a purification problem. So to purify the protein, we use immobilized metal ion chromatography, and gel filtration. And we also test adjuvant effectiveness according to various adjuvant. When you inject this adjuvant, ASO3 and MF59 seems to be a high PRNT level. So we use ASO3 as adjuvant. This study was supposed to finish until September due to project timeline. So we'd like to use the interferon knockout mouse, but there is no interferon knockout mouse available. So we have to use interferon receptor blocker. But we don't know the receptor blocker dosage. So after Google it, we use 0.1 milligram per mouse. Inoculum size, challenge size 10^3. This is a PRNT level before SFTS challenge. The level is satisfactory. When you check the body weight, this black line is vehicle group, vaccine group shows higher body weight. But the problem is, even though vehicle group, which is just inject saline, 100% mouse will survive. As you know, when you challenge virus, mouse have to be dead so as to check vaccine effectiveness. But it is not impossible to check vaccine effectiveness. But when you checked viral copy number in spring, it is very interesting. When we inject MP group, viral copy level is lowest. As you know, MP did not release neutralizing antibody at all, but MP is already known induct cell-mediated immunity. So I have hypothesized if we made MP engineering, it can be very good antigen. So we use engineering NP protein using cell-penetrating peptide. If the NP antigen uptake in cellular mode, it can be more effective in cellular immune response. So we made a variety of engineering NP protein. NP6 seems to be best. So we checked intracellular uptake by confocal microscope. NP1 is original NP. As you can see, NP6 seems to be the best. So we'd like to check higher intracellular uptake correlate with cell-mediated immunity. So I checked interferon gamma level and TNF alpha level. As you can see, NP6 seems to be best. And in America, there is a similar SFTS virus. It is called heartland virus. So I make heartland NP protein. So when we stimulate with heartland NP, it can also elicit cell-mediated immunity as well. And so we performed third experiment. At this time, we use interferon receptor blocker to 200 microgram, and then we higher the virus challenge to 10 to 5. As you can see, this is a vehicle group. This is a plant GM plus NP. This is a plant NP. This is E. coli NP6. So all vaccine groups survive. And body weight graph is also good. And this is, as you can see, MP6 group has a low viral copy number as well. This is a mammalian cell vaccine study. Red one is MP injection group. This one is GN, this one is GC. As you can see, our E. coli MP6 seems to be better than the mammalian cell vaccine group. This is a pathology study, mark group and virus group. When you challenge the virus, a white bulb seems to be disrupted. In kidney, glomerulus messenger cell infiltration was detected in virus group. And in liver, there is a coagulation necrosis detected, but in vaccine group, it is not that evident. When you're making it a scoring system, as you can see, MP6 seems to be better. Glomerular injury was less in MP6 group. And we also made mini-body antibody. This is IgG, intact IgG. This is SCFV. SCFV is variable region of light chain linked to variable region of heavy chain with a linker. Our developed mini-body is this one, like this. So we collect PBMC from SFTS recover patients to make cDNA. So we made human immune mini body using human immune library through panning. So we made 4 mini body. Among 4, 3 of them has good neutralizing activity. So we do challenge tests. This mini body, we inject only 1 dose. Just this is SFTS injection time and 1 dose of mini body. This group inject 4 consecutive days. And isotope, it has a mini body against leptospira, and the other is against SFTS virus. Survival curve, as you can see, mini body injection group all survive, even though just 1 injection all survive. As you know, we first challenge the virus, and then we inject mini body. So at first, the body weight tends to decrease and rapidly recover in mini body group. When we make scoring system, mini body group, both of them is very effective. So that is all I prepared today, and most of the company did not interest in SFTS virus because it is very low prevalences. So I hope our developed mini body and vaccine can be available, can be used in clinical setting. Okay, thank you very much.
Well, many thanks, Professor Kim. That was a fascinating presentation. Can I go back to the beginning, which is about the virus natural life cycle, perhaps? Do you know if there is an animal host for SFTS virus? Because we see with other tick-borne infections, the tick normally infects other animals rather than humans. Do we know anything about that at all?
Yeah, goat, lamb is the host, and there is a companion animal can be infected with SFTS virus as well. Especially cats are very susceptible to SFTS virus, and dog, well. So a lot of animals can be infected with SFTS virus.
Right. And do we have an understanding of rates of subclinical infection with SFTS? Obviously, a PCR test will only pick up replicating virus, but what about serological surveys? Is that possible, and does that show anything?
Yeah, actually, when you perform the serologic test using IFA test, in Korea, around 4% of people have seropositive rate. In China, it also has 4% of positive, but in Japan, the prevalence is less than 0.4%, around 0.2 to 0.3%. Yes.
So the severe disease is a relatively small proportion of total infection. Is that correct?
Right. If the patient is healthy, in that case, as you said before, there is some asymptomatic infection as well. Actually, my research worker has a tick bite, and we perform, when you go to the KCDC to do experiment to make IFA, we check that IFA level, but IFA level is more than 10,000.
Right.
So, yeah, asymptomatic very common.
And is human serum then a possible treatment for patients with severe infection?
Of course. And especially there is some data using recovered patients. Plasma sample were used. In Korea, one of the Korea hospital, they report the use of plasma therapy after recovered one. It can show effectiveness, especially when the patient receive that plasma, viral copy number decrease sharply and antibody level increase a lot. And so we can use recover the patient's plasma. But when it comes to IV pharmaceuticals, in IV, as you know, it is a new infectious disease. So those kind of IV pharmaceutical, they don't have those kind of SFTS antibody.
Yes.
So it does not seem to be effective. But if the patients recover from the SFTS, in that case, we can use those serum. But it is not easy to use this one.
I was fascinated to see that post-infection mini-bodies were seeming to show promise for effective treatment. They work very quickly then.
Yeah. Actually, it is a monoclonal antibody.
Yeah.
Kind of monoclonal antibody, but it is just a mini body. So the size is smaller than IgG.
Yeah.
And it can be made with low cost as well. And so it can be very useful if it is used in clinical setting. So I hope we can perform clinical study.
Absolutely.
Thank you for presenting such a fascinating lecture on SFTS. I will now introduce my colleague, Professor Linfa Wang. He is a professor in emerging infectious disease at Duke and US, and he's also the executive director of Singapore's pandemic research program called PREPARE. And he has played a key role in the discovery of Hendra virus, tracing SARS-1 to bats, as well as the discovery of Langya virus in China recently. If you describe what he is, he's a expert in beta coronaviruses, and which is very, very topical. And they call him the Batman. And he will now share with us about Nipah virus. And I'm sure he'll talk about the role of bats as well. Over to you, Linfa.
Great. So the topic is on Nipah virus, and because the theme is on one house, so I put a subtitle. Really, so use that as an example of one house approach to emerging infectious diseases. Yes, David. Of course, Nipah virus. You have to talk about emerging bat viruses, and then expand from Nipah to Hendra, and then the genus we call the Hannipaviruses. And in that genus, we have many newly discovered viruses I will just briefly describe. And then I think to summarize, the international effort, because Nipah is on the WHO's priority list. So internationally, we're working very hard, try to get, just in case Nipah become more transmissible. So we have the Nipah virus sort of outbreak preparedness, effort. And then, come back to one house. We finish the talk with a one house approach to outbreak prevention and vaccine development, basically. So this is 25 years, 94, to now, and so it's a quarter of centuries studies on emerging backbone zoonotic viruses, and I'll always consider myself fortunate because I was not trained as a virologist, but I went to Australia. And I went to the Institute 1990, and we discovered this Hendra virus 94. So from Hendra 94 to Nipah, and then SARS-1, MERS, coronavirus, Ebola, and COVID-19. All of them, I think, are believed to be, backbone virus. At least their ancestral viruses are coming from bats. So I was maybe the only one internationally was fortunate enough to be involved in studying all these 6 different viruses. And clearly that, they will not be the last one to emerge, right, COVID-19. And also it can happen anywhere. So I always say the first really deadly virus emerging in a hantavirus from bats happened in the least likely place in Australia. Hendra is a suburb of Brisbane. Australia, as you know, is an island country, very low density population and animal-human interaction. We don't have a live animal market. And yet the first emerging zoonotic backbone virus really happened there. And then the Nipah virus, of course, the Nipah, the first discovery in 98, 99 in Malaysia and Singapore. And that was followed by the SARS-1, Southern China. And then we had the MERS coronavirus, Middle East. And then we have Ebola virus, West Africa. And then we're back to Central China for SARS-CoV-2. So basically, as I said, these are all originated from bats long time ago, and then can happen anywhere, basically. So Nipah virus was, as I said, the first discovery was in 1999, and for a major outbreak disease, in pigs and people, in Malaysia, and spill over to Singapore. And result, in many hundreds infections, and the case fatality was like 40%. In order to control the outbreak, the government have to cow, more than 1 million pigs. So there's a huge economic impact, just like COVID-19. So for those of you interested in history, I would strongly recommend, this a personal kind of journey by Dr. Cao-Bing Charles that's him. So he really single-handedly discovered that Nipah virus. And it's a very interesting story. So for the young scientists in the audience and the clinicians, because he was a clinician and working in the diagnostic virology lab. So for any of you interested in this kind of journey, I really strongly advise you to read his personal account. Now, sort of if we look at internationally, so these are the areas that we already have human cases of Nipah virus infection. Of course, Nipah infection largely lethal, so means there's also death. So as I said, it started here in Malaysia, Singapore, and then we have Bangladesh, India, and we have in the Philippines, and more recently in India. Now we have on the West Coast. So again, these are only the detected and reported human cases with fatality, but there is a possibility, historically, there might be other outbreaks that we have missed. So if you look at this, from the Ministry of Health from Bangladesh, so Bangladesh now is the country really most affected. The first detection was 2001. And as you can see that, so these are the cases in dark blue, and then the fatal cases in light blue. And the case fatality rate, that's the most scary part, right? It fluctuates, but it can go as high as the 100%, average around 70%. So this is really the most deadly virus. And most people are aware of Ebola virus being very deadly, but really Nipah is even more deadly, and it's claimed to be the most deadly virus that we human beings have ever been exposed. So we have to bring in the Hendra virus, right? Because the Hendra was like a close cousin, and Hendra is a zoonotic virus first found and only found in Australia. So first isolated in 94 after a major outbreak of horses, and only 2 humans were infected, but 1 died. So it's a 50% case fatality. So it initially was named as an equine sort of mobility virus because it's from horse and the closest to measles virus, a mobility virus. And I was responsible to rename the virus as a Hendra virus, depends on the genome and things like that. And then the virus has been, connected to many sort of deaths of domestic horses. But in terms of human, that only 7 cases. So Hendra and the Nipah, when I was, started to look into the genome, I discovered that these are paramyxoviruses, viruses, but that's not really grouped with any of the existing genera at that time. So mobility virus, respiratory virus, and the Ebola virus, at that time, only 3 genera. So we decided really to say, go to the international committee to suggest that we need a new genus to accommodate these new viruses, and we proposed the name Nipah virus. And that, 2 years later, that was officially approved. So now, for those of you, the virologists, the fused virology is like the Bible for us, right? So we were very privileged, the fused virology actually create a separate chapter for the genus Hannipaviruses. So now, from now on, we were referring to Nipah, Hannipavirus, hantaviruses, all in the same context because these viruses are very similar. They are paramyxovirus virus in the family. So paramyxovirus is enveloped virus. You have the membrane there, and they have two major surface proteins. One is a fusion protein. The other depends on the activity called the HN or just glycoprotein. And then inside there, you have the matrix protein there, and then it's a negative strand non-segmented RNA genome. And that RNA genome, because it's a negative sense RNA, is never naked. It's always wrapped in a nucleic capsid with the polymerase, the large protein, the phosphoprotein, and the nucleic capsid protein. And then for Hendra and the Nipah, the genome is really big. Before we discovered the Hannipavirus, the genome is all less than 16 kilobase, and the Hendra is over 18 kilobase, and that's also is a significant discovery. So this is the current sort of taxonomy. The Paramyxomyridae, the family now is divided into 4 different subfamilies. And also Myxovirinae, the subfamily is, I think, the most important in terms of human, pathogens, because, we have the measles virus, the human parainfluenza virus, all in that subfamily. And now, instead of 3 genera, when we discuss the Hannipavirus, now we have 8 genera, and the Hannipavirus is one of them that I sort of to help to establish. In terms of the species in that genus, now there are, 6 which are recognized, so the Sida virus, the Ghana virus, Hendra, Nipah. Of course, the prototype is Hendra, and with Hendra and Nipah, we named the Hannipavirus. And then we discovered the Zhejiang virus and the Lanya virus. So these are ICTV recognized, but in terms of the viruses that we have a genetic sequence, we have many more I will show you later. So clearly, as I said, these are viruses that are using bats as the reservoir for the ancestral virus. But during the evolution, some of these bat viruses will spill over to other, animals, like pigs and horses, or directly transmit to human through, fruits, the eating fruits, and the fruits can be, half eaten and dropped into the pig pen, or, bites and scratch. So there are many different ways they can transmit to human, and then eventually that some of them have human-to-human transmission. Luckily, the Hannipavirus are so far not very transmissible, but if any mutation makes this virus transmissible, then I think we will be in a big sort of a disaster. That's why WHO put this onto the priority list. So this is the current list, just updated by WHO. So before of COVID-19, Nipah was number 3, but now COVID-19 is number 1. And then Crimea Congo hemorrhagic number 2, Ebola, Lassa, MERS, and SARS. And then the Nipah and the Hannipaviruses are there, still considered as a top priority pathogen for potential future outbreak. So now this is the updated phylogeny. So if you look at Nipah is here, Hendra is there, because we have now Nipah is 2 sort of varieties, Nipah Malaysia, Nipah Bangladesh. Hendra, although it's only limited in Australia, now we have 2 different genome types, the original Hendra and Hendra genotype 2. And then we have many these viruses, and the host range now is go beyond bats. We have Hannipavirus from rats, we have Hannipavirus from shrews, but only 3 so far have demonstrated ability and cause human disease. Of course, the Hendra and the Nipah, the classic Hannipavirus, as I said, they are highly lethal. Case for the fatality ranges from 40% to 100%. And then David mentioned, introduction, we recently discovered this Lanya virus, so it's a very close cousin of the Hannipavirus. Because it's new, so many of you in the audience may never heard of this, so I thought I'd just show you a few slides, So this was published now more than a year ago. And so basically it's a zoonotic, we know, and a hantavirus, and in febrile patients in China. So this is a collaboration with the Beijing Institute of Epidemiology. The study design was very simple. Basically covered the period from April 2018 to August 2021. And then really in 3 hospitals in 2 provinces in China, the Shandong and the Henan province. It's lit up on the east side. So the selection criteria is only 2. Patients with acute fever, 38 degrees or above, and they have to answer the first question. Have you had a contact with an animal, any animal, your pet, your livestock, your wildlife, 1 month, within a month of disease onset? If you said yes and yes with these 2 criteria, either inpatient or outpatients, we recruit them. And we take a blood and the throat swab, and with, of course, a questionnaire and a clinical data laboratory test, if they have a test, and also if they were under treatment, and then the treatment details. But importantly, we also conducted a community-based investigation on these apparently healthy individuals living in the same area and also during the same study period. Because that's very important. These are our controls, to see if there have been any sort of asymptomatic infection or past exposure in that population. Okay, so, the study was so successful, within a few months, we identified the index case. So this is a 53-year-old female from the Langya township outside of the Qingdao city. That's why we call it the Langya virus. And she was suffering from fever and many different, symptoms, and was, was severe enough to be admitted to hospital. So it's in sort of a patient. So from there, took the sample, and the laboratory test again, really indicate that she's suffering some of the clinical abnormalities. But the test, in terms of etiology, whether it's a bacterial virus, it's all negative. So now it's a pretty standard, approach, right? If you could not find any pathogen, you go for a metagenomic analysis. So from there, the metagenomic unbiased sequence revealed that that patient actually has a very high level of the viral RNA. So we were able to get just, a non-targeted sequence, it's almost a full-length genome. And the virus is very close related to the known Bandaviruses. And then we did some further investigation. So in this cohort, we had a total of 1,600 febrile patients, and we tested by PCR for this Langya virus. And surprisingly, the index case was not the only one. So we have around 35 were positive for Langya virus. And then from the healthy individuals, because at that time they did not display any symptom, 4 of them were positive for Langya virus. So you can argue that there are some asymptomatic infection. But what's really surprising is that 2% seropositive in that population in terms of the IgG antibodies. And then of the 21 positive healthy subjects, we asked, profession and also, their past experience of animals, and all reported history of contact with animals. And I don't have the time to show you the data, but you can read our paper. Basically, we have presented evidence to suggest that shrews are the most likely origin of the Langya virus and some related viruses. So basically, we have demonstrated that a brand new virus jumped to human. Maybe it's not the first time. It causes diseases that's from severe pneumonia to mild sort of respiratory symptom to maybe asymptomatic. And there was we also investigated house contact and things like that. It seems like there's no human-to-human transmission. So basically, a virus is not ready for human-to-human transmission, but frequently jumping to humans and can cause disease. So I think to get asked, that's a new virus, should we be worried about Langya virus? And I says, It's no and a yes. The big no is because so far there's no evidence of human-to-human transmission. But I don't think we can totally discount that. As we know, the virus can mutate. And what caused the concern of we should be worried about the although it's a tiny yes, but because this lung virus is sort of phylogenetically related to Hannipavirus or Nipah, and also measles virus. I already said Nipah is the most deadly virus that we ever been exposed for human. Measles is the most transmissible virus, right? We human have been exposed. So if you have a virus, related to both of these viruses, or you need a few mutations, it can cause a major sort of issue. So I think we should watch out. All right, so now I switch to the topic on Nipah virus outbreak preparedness. And there are three areas like the vaccine, therapeutics, and the rapid point of care test. So, I don't have the time to go through data and things like that. Just a high level summary to say, scientifically, research-wise, many activities, and then we have, research products for vaccine, therapeutic, and tests. But if you focus on the sort of licensed products, very few in all three categories. So CEPI, which is the Coalition for Epidemic Preparedness Innovations, have recently, in the last maybe few years, funded the 4 individual Nipah virus vaccine projects. So the first one is by Vaccine in USA, actually use our Hendra sort of soluble G vaccine, because the Hendra and Nipah cross-protect almost with 100% efficiency. So this is a protein subunit vaccine, and now is in human phase clinical trial. And the second is a public health vaccine, is a recombinant VSV based. And third one from University of Oxford is the ChEdOx, the same vector used for the AstraZeneca vaccine. The last is from University of Tokyo, and it's recombinant measles viral vaccine. So it's a 3 viral vector, and then 1 is soluble subunit protein. And clearly now there are companies now go into develop mRNA-based Nipah vaccines. So different from COVID-19 or coronavirus, Nipah virus is genetically very stable, and the protection is, the correlate of protection is neutral antibody. So although the disease is deadly, but the vaccine, I think, is highly sort of effective, and I think we can achieve, immunity against infectional transmission for Nipah virus. In terms of therapeutics, of course, there are small molecules and monoclonal antibodies. Again, I don't want to do an extensive review, don't have the time. Just give you one example that I was involved. We developed a monoclonal antibody against the Hendra virus, highly potent. David, you were asked, the question was SFTS. So this antibody, if you give before infection, any model is fully protective. If you give within 2 to 3 days after infection, they still offer some protection. So the antibody, now is already gone through phase 1. Lastly, in the rapid point of care POC test, so CEPI just funded around $15 million, new initiative, approved in February 2024, target 2 viruses, Lassa fever and Nipah virus. So we are sort of optimistic, we will in the next few years, we will have a rapid test for both antigen and antibodies, and just like the one we use for COVID-19. So I finished my talk on this novel approach, sort of to do a One Health vaccine approach. So these are the known kind of Hannipavirus outbreak and how they transmit and the case fatalities, right? So the first was Hendra in Australia in '94, and then, and Nipah Philippines, it's all through horses to human. And the Nipah Malaysia and Singapore is through pigs, and there's a massive pig-to-pig transmission. And Nipah in Bangladesh and India, we believe is a bat-to-human transmission, not directly make a contact with human, but through contaminated the juice, palm juice. And so we, when I was still working in Australia, we developed this first vaccine against Hendra virus. So we developed a vaccine against Hendra. Of course, you want to protect the horses, but the key aim is to block that transmission, because as I said, the Hendra and the Nipah vaccines are very effective. Neutral antibody is correlates of protection, and they actually can prevent infection and transmission. So this is and we got a licensed as a vaccine for horse use. So it remains the only vaccine, that licensed vaccine for a biosafety level 4 agent. And the same vaccine was very effective for Nipah virus. So that's why CEPI founded, for a human clinical trial. Since this vaccine was applied, in Australia, hundreds, thousands of, doses, not a single horse, vaccinated horse, has been infected by this hantavirus, whereas non-vaccinated horses, every other year we still have a few cases. So really, in the real sort of world, also demonstrate efficacy. All right, so just to finish up, to say, this is an emerging virus area, but we have been going on. Nipah was discovered, 1999. So just before COVID-19, December 9 to 10th, I organized an international conference called the Nipah at 20. Now, Hanja was discovered in 1994. So this year, I'm a co-organizer, I'm on the committee for the Hanja at 30. So it will be held in Australia, where the Nipah hantavirus was discovered in the lab. So this is the Australian Animal Health Laboratory. Now it's called the Australian Center for Disease Preparedness. And the Geelong is a very beautiful sort of city, and hopefully some of you in the audience can attend. Thank you.
Thanks, Linfa, for a very good primer on Nipah viruses. There's a question from an audience member, Salim Zaman. He observed that Nipah infection occurs in winter in Bangladesh, summer in Kerala. Do you have any idea about that, or is it weather independent?
Yeah, so this is a seasonality, right? I think that in Australia, for example, we had many cases of this. So we have been doing that. And we don't find that in terms of viral load in bats, there's really no seasonality. But in India, that I'm not sure whether that's due to the virus secretion or due to the practice, the social practice of the community. So usually we say that there's no seasonality for the virus cycle in bats, but I don't know if the human infection are influenced by other factors.
Okay, any other question for Professor Linfa Wang?
Well, that was a fascinating presentation, and it reminds me, back in 1984, when I was a medical student, I spent a couple of months in Mauritius, and we met someone who was doing captive breeding of Rodrigues fruit bats. So these are bats from an island way in the Indian Ocean, isolated from everywhere else. And because I didn't know any better, he said to me, "Do you want to go and look at them?" So I was in the cage with these bats, and they were flying around me with these bits of oranges and things hanging out their mouths. And I possibly had a close escape there from catching a virus of one sort or another. But it makes me wonder, I mean, Rodrigues is very, very isolated, and Mauritius so as well. Have you done surveys of fruit bats around the world and looking for whether these various viruses that are causing human diseases, are they distributed unequally around the world in fruit bats? Is there do some bats not have them?
Yeah, so equally, I think it would be an overstatement, but certainly bats are very ancient mammals. They diverged from land mammals 65 million years ago. So we always say that no matter where the bats are, and also I don't have the time, of course, to go to the bat immunology, because the bat immunology tells us that bats are ideal reservoir for all sorts of different viruses. So I think one sort of a debate whether bats carry more virus per mammalian species than other mammals. The answer is yes. I think that it's being resolved. So, but not all bats carry all the viruses. That's for sure. So the geographic location and isolation, as you say, on the island. But the other thing is biology. Clearly, like the coronaviruses, in the Rhinoceros genus, but certain species carry certain viruses. So we still don't understand that virus-host interaction, the regime factors for certain viruses. Yeah. So overall, bats carry more viruses than other mammals, but not all bats carry all viruses.
Yeah, wonderful. And the immune protection that bats have then presumably is of interest.
Yeah.
How do they protect themselves from the pathology of these viruses?
Yeah. So that's now it's 80% of was my research focus until COVID. So now I'm switching back. So, so I can send you a article we put in Nature. Basically, is about the defense versus tolerance.
Yes.
So for us, our interferons, cytokines, humans are so adapted to a safe, clean environment. So we don't switch our defense system. But any virus infection, we will fight really hard. And eventually you have the pathology or death. So virologists, we have a cliche say, very few virus kills us, we kill ourselves, right? It's our immune response.
Yes.
Bats, on the other hand, as a flying mammal, they're constantly bombarded by this kind of stress, DNA, oxidative stress, and the heartbeats go to 1,000 beats, body temperature goes to 42 degrees. Every night when they fly, they have this stress, okay? So they have developed a system that is much more balanced to fight against the stress and then tolerate. And then infection is just part of the stress equations, right? The signaling pathways, for interferons are shared by, DNA damage and infection. So that's why bats actually developed a system to say, I'm going to live with you. I don't fight. And that's why they don't cause disease.
Amazing.
So it's actually, it's not, they clear the virus, but they can tolerate the virus better.
Wonderful. Many thanks indeed. I think we're out of time now, David, I think, aren't we? So we'll just finish off. Well, I'd like to personally thank all three speakers for a fascinating session. I've personally learned a lot this morning about a wide range of diseases and antimicrobial resistance and control. So I'm very grateful to your time and effort. David, do you want to say anything at all? Finish off, because I just want to put one slide up. I
share your sentiment. It is really interesting. And this also shows the collaboration of APSCMI with ISAC. And of course, we all want to go to Istanbul. It is where the East meets the West. And it will be happening on the 3rd of November this year. Many of us will be there, and we hope to see most of you there in Istanbul.
Many thanks. Excellent.
Just like this webinar, where the East meets the West.
So thank you very much, everyone, for joining, and I hope you found this as educational and entertaining as we did. And we look forward to seeing you in Istanbul, but also to the next webinar. Keep looking on the ISAC website for the next webinar. Thank you very much indeed.